Hi everyone, so let's continue on. So we talked about post-translational modification of proteins just a little bit, and so there's these reversible covalent modifications that can help turn on and off different pathways inside the cell. Now the prototype in eukaryotic cells is protein phosphorylation. Protein phosphorylation is probably and I think yes, it is the most common modification that can happen to proteins. Protein phosphorylation. This happens approximately 70% of times inside the cells. So protein phosphorylation is an extremely common event that happens inside our cells, and it usually occurs on the side chains, the hydroxyl side chains. So if we're talking about hydroxyl side chains, we're probably talking about tyrosine, serine, and threonine. Okay, so most common with these three amino acids, and it is a prototype. But there can be other kind of modifications too, just like I mentioned before, methylation. And when you hear the word methylation, you should think about DNA methylation, right? I think we all have learned that at a very early stage in our biological careers. So DNA methylation. Then we have acetylation. You can add acetyl group, so that is referred to as acetylation. You can have ubiquitination, you can have simylation, and so on and so forth. So there's many different kinds of modifications that you could do to your proteins. Now, just an extra point that we should mention here is that you can have a protein. So let me draw my protein. So I have this protein here. So I can either phosphorylate or modify this, or let's say phosphorylation. So I can phosphorylate this protein at different sites. So it's not that you can only phosphorylate the protein chain, the peptide chain, at only one places. You can phosphorylate it at many different places, right? Depending on wherever your hydroxyl or whatever amino acids you need for that particular modification. So you can do that, or you can have your protein and you can modify it with different modifications at different places. So either I can have a methylation here, I can have a phosphorylation here, I can have an acetylation here, I can have a methylation again, I can have a like anything is possible. You can have a limitless set of different modifications on just one single peptide, on one single protein. So that is one point that I needed to make here. Now, what can covalent modification of proteins do? Okay, let's discuss those now quickly, and hopefully I can finish those off in this video itself. So say I have a protein. So the first thing that they can do is they change the location of the protein. Now how do they do that? Now say I have this protein lying around in the cell. Without that modification, this protein cannot enter the nucleus. Okay, now as soon as I modify it, that modification sort of acts as a ticket for this protein to enter the nucleus. So with that modification, it can enter the nucleus. So this modification has pretty much changed the location of my protein. What it can do next, it can alter activity. So I have this protein, and this is involved in, let's say, biochemical pathway A, whatever that pathway is. Now if I modify it, so if I add a modification to it, it will no longer act on this pathway. So I have changed its activity, I have changed the structure, changed the structure in the sense of modified the structure, I've changed its activity. It can provide a specific interaction domain for other proteins or molecules. So I have this one protein here, and I have another protein here. Now these two proteins, they have no affinity for each other like this. As soon as I add a modification to it, this protein recognizes it, and this protein has an affinity for it, and they both sort of combine and interact. So this modification on the blue protein pretty much created an interaction domain for the yellow protein, and yellow protein to come interact with it. And they can control proteolytic degradation. So I have this protein. If I add a modification to it, that can signal the cell to degrade it, like it's not needed anymore, it's not functional anymore. So that modification can act for its degradation. So it can signal the cell that, okay, here's this protein, please degrade it, we don't need it anymore. Okay, so that is the last function of the covalent modification of proteins. So I'll end this video here. There's 3 seconds left, and in the next one we'll start with an example of protein